Bismillahirrahmanirrahim, and welcome back to this next video. This is another video in the series of videos on the eosinophils. We were talking about the granules and the components of the granules in the eosinophils, and in this particular video, we are going to see another important component that is present in the granules of the eosinophils, and that is known as the major basic protein. Now, this major basic protein, it is present in the granules of the eosinophils, and this is the predominant constituents in the eosinophil granules. If you talk about the major basic protein, they contain high content of the basic amino acids such as the arginine and the lysine, and because of the presence of this high content of the basic amino acid, the charge on the major basic protein, that is positive. Now, this positive charge, it helps the major basic protein to bind to negatively charged molecules like the phospholipid, which is actually the target of the major basic protein. And this interaction between the positively charged amino acid in the MBP and the phospholipid, which is present in the target cells, the MBP is actually going to achieve its function. As the major target for the major basic protein, that is the phospholipid. So let me give you a brief overview of the structure of the phospholipid. Now, if you look at the phospholipid, we have got a glycerol molecule over here, and to the two carbons of the glycerol, you have got two fatty acid attached to it. The third carbon, that actually have got a phosphate group, and because of this phosphate group, the charge on the phospholipid, that is actually negative, and the basic amino acid which are present in the MBP, they are interacting with these negatively charged phosphate group present in the lipid bilayer in these phospholipids. Now, if you look at this phospholipid, this particular head, that is hydrophilic in nature, and these tails, which are actually the fatty acids, they are hydrophobic in nature. And because of the presence of the phospholipid, the membranes, they are going to achieve their integrity, and they are going to achieve their semi-permeable nature. If you talk about the molecular weight as the major basic protein, that is actually a family of the protein. So the molecular weight is between 14 to 16 kilodaltons. Uh, let us have a discussion on the mechanism of action of the MBPs, how they work. If you talk about their mechanism of action, so this MBP is a very potent enzyme against the helminths, and it is toxic towards the bacteria and the mammalian cells in vitro. If you look at these helminths, for example, one of the important example from the helminths family, that would be the roundworm. Another important example, that would be the typeworm. And these helminths, they are responsible for the parasitic infections in the mammals, for example, including the human beings. Now, the major basic protein, what it also do is that it is responsible for the release of the histamine from the mast cells and the basophils, and it's also responsible for the activation of the neutrophils and the macrophages. But we are going to focus on the MBP as a degrader or as a destabilizer of the membranes of these roundworms or the tapeworms, for example, in this particular video. Let us have a detailed discussion on this. If this is the major basic protein, for example, this one is your lipid bilayer. This is one phospholipid, another phospholipid. You have got a lipid bilayer over here. So what happens is that the MBP, that is going to interact with the membrane, as you can see in this particular image. These one are the phospholipids of your membrane, and this one is the structure of the MBP. So the MBP, what it do is that it is going to interact with the membrane. It is going to insert itself into the membrane, and what this insertion do is that it is responsible for the permeabilization of the membrane. As you can see over here, I've got an intact lipid bilayer over here, and when the MBP it insert itself into the membrane, that is going to create pores in the membrane or in this particular lipid bilayer. So the end result of this membrane permeabilization is that it is going to increase the permeability of the membrane. That means that the semi-permeable nature of the membrane that is gone. What you do is that when the membrane permeability that is lost, that is going for the leakage of the cellular contents, and when there is leakage of the cellular contents, that actually leads to the cell lysis or the death. So, for example, if this is the lipid bilayer of the roundworm, for example, at the end of the day, the MBP is going to increase their cellular permeability. That is going for the leakage of the cellular content, and that is going to lead to the cell death of the roundworm, for example. We will continue the discussion in the next video.